Okay, so when we left off on Tuesday, we were talking about the structure of the DNA molecule. Okay, so specifically, we were talking about the interactions of the bases. So remember the bases. Well, first of all, remember DNA is a double helix, and it has it's a double-stranded helix composed of two anti-parallel strands. So remember, there's a polarity to the DNA molecule. It goes from 5' to 3'. So we've got one strand running in the 5' to 3' direction one way, 1.5' to 3' the other way. Okay, so when the bases are lined up anti-parallel, that's when the proton donors and acceptors in the base pairs line up so that they can form base pairs. Okay, so we can get we get 3 hydrogen bonds in a GC base pair. We get 2 hydrogen bonds because we have 2 proton donors and acceptors lining up when we have an AT base pair. Okay. So also these base pairs stack very neatly upon each other. Let's go back to a nice picture of the helix. Okay, so here, particularly on the left-hand side of this figure, you can see how neatly the base pairs stack on top of each other. And base stacking interactions are another interaction that stabilizes the DNA double helix. Okay. Another one of the forces that stabilize the DNA double helix are what are called base stacking interactions. Part of these base stacking interactions are what are called pi-pi interactions. So these are electron interactions between stacked bases. They also contribute to the stability of the helix. So remember, in these bases, we have these ring structures. The ring structures contain pi bonds, and those pi bonds contribute to electron cloud interactions called pi-pi interactions that stabilize the DNA double helix. So this contributes to base stacking interactions that stabilizes the helix. Okay, certain enzymes, such as DNA methylases, DNA repair enzymes, can flip single bases out of the helix. So this is kind of cool. Let's talk about one enzyme in particular that's found in E. coli. There's an enzyme in E. coli called DAM methylase, and it methylates GC base pairs. So you might be thinking, in order for this to happen, you would think we'd have to melt the DNA double helix, okay, which is very thermodynamically unfavorable, in order for the enzyme to get access to the bases to add methyl groups to the bases. Okay? When scientists studied DAM methylase, what they found was instead of melting the DNA double helix in order to get access to the base, just take the base and flips it out of the helix like this. And you can see that happening here. So just kind of takes one base pair, flips it out like that. Okay. Now I have a question here. Do you think this is thermodynamically favorable? Flipping out like this, flipping a base out. Okay, so in order to do this, we have to upset the base stacking interactions. We have this very negatively charged phosphosugar backbone on the outside interacting with water. We have a pretty hydrophobic environment in the center. So this actually isn't very thermodynamically favorable. Okay. However, it is more thermodynamically favorable than melting a local region of the DNA in order to get access to the bases. Okay, so it's kind of again, it seems like in biology, evolution finds the most elegant solution to the problem sometimes. And this is an elegant solution to the problem. Just by flipping out a single base, it can be modified. In the case of DAM methylase, it can be methylated. There are some DNA repair enzymes that can repair a base and then flip it back in as well. All right, most DNA is a right-handed helix. The way that we can tell a right-handed helix from a left-handed helix is by something called the right-hand rule. Okay, so if you look at, I wish I had a model of the DNA molecule to show this on, but you're just going to have to kind of imagine this in 3 dimensions. Okay, so if you can imagine taking the fingers of your right hand and running it along the edge of the helix in the forward direction like this. If it's a right-handed helix, your hand will move in the same direction that your thumb is pointing. Okay, on the other hand, if it's a left-handed helix, then your thumb is going to be moving in the opposite direction. So here's a right-handed helix. So if you can imagine just taking your fingers, running it along. Let me move the camera so that the people zooming in can see this. Okay, this might be really difficult to see, but in fact, it's impossible to see. So let me just go back to the PowerPoint. I'll just describe to the people joining by Zoom what I'm doing. Okay, so if you look at the helix over on the right-hand side, so imagine taking your right hand and running your fingers along the outside of the helix. Okay, if your thumb moves in the same direction, if your hand moves in the same direction that your thumb is pointing, then you have a right-handed helix. Okay, now obviously that wouldn't work with this left-handed helix. I ran my fingers along it, my hand is moving in the opposite direction, my thumb is pointing. So this is a left-handed helix. Okay, so what's described there is called the right-hand rule. We use the right-hand rule to distinguish a right-handed helix from a left-handed helix. Okay, so some characteristics of the DNA double helix. DNA has a periodicity of 10.5 base pairs per turn. Each base pair in the stack is twisted about 36 degrees relative to the one before. So we all know that there's 36 degrees in a circle. So if we twist each base pair about 36 degrees, then 36 times 10 is 60. So we have about 10 base pairs per complete turn of the DNA helix. Okay, DNA has major grooves and minor grooves. And the reason we have major grooves and minor grooves is because the angles of the glycosidic bonds joining the bases to the 5-carbon sugar deoxyribose. Okay, let me back up and find a nice picture of the helix to show you the major and minor grooves first. Okay, here's a good picture. Okay, and even so, the major grooves and the minor grooves, well, the minor groove is labeled. So we can see, here's a major groove. So we can see a major groove is very deep. On the opposite side of the major groove, we have a minor groove, which is very shallow. Now, the reason that DNA has major grooves and minor grooves, again, has to do with the angle of the glycosidic bond that joins the 5-carbon sugar to the nitrogen-containing base. All right, so let me get my drawing tool back. So those glycosidic bonds that I'm talking about are here and here. Okay, so these glycosidic bonds join the base to the deoxyribose at the 1' carbon of the deoxyribose. Okay, so if you can imagine, like, imagine this base pair like at the center of a clock. So the minor groove goes here to here, okay, from about 8 o'clock to about 4 o'clock. Okay, then from this glycosidic bond all the way around to the other one. So from about, let's call that about 8 o'clock to about 4 o'clock in the clockwise direction. All of that's the major groove and the minor groove. Okay, same thing down here. So this glycosidic bond to this glycosidic bond, that's the minor groove. Then here all the way over to here, that gives us the major groove. Okay, does that make sense? Okay, the information in the minor groove cannot distinguish a GC base pair from a CG base pair, nor can it distinguish an AT base pair from a TA base pair. Okay, so the information is in the form of proton donors and acceptors on the edges of the base pairs. The minor groove also cannot accommodate many amino acid side chains, whereas the major groove can fit an alpha helix nicely. Okay. So let's look. So like I said, the information I'm talking about is in the form of proton donors and acceptors. So if we look at the information here, so let's look at the major groove. So in a GC base pair, we have a pattern of proton acceptor, proton donor, and then a hydrogen. Okay, over here, we have a pattern of hydrogen, proton donor, proton acceptor. So DNA binding proteins can recognize these proton donor acceptors and hydrogens by their pattern. So we have a different pattern for a GC base pair than we do a CG base pair if we're looking at the patterns in the major groove. Okay, in the minor groove, this information is ambiguous. Okay, so look here. Let's look at the information in the minor groove. We've got proton acceptor, proton donor, proton acceptor in a GC base pair. If we look in a CG base pair, we have the same pattern of proton donors and acceptors. Acceptor, donor, acceptor. Okay? So this is why proteins can't tell the difference between a GC base pair and a CG base pair if they look at the information in the minor groove. Okay? The information is ambiguous. Okay? However, if it looks in the major groove, then they can be distinguished from each other by the pattern of proton donors and proton acceptors. All right, look. Let's look at an AT base pair. So again, in the major groove, we have proton acceptor, proton donor, proton acceptor, methyl group. If we look at a TA base pair, methyl group, proton acceptor, proton donor, proton acceptor. Okay, so the patterns are different, so we can distinguish an AT base pair from a TA base pair if we're looking at the information contained in the major groove. Let's look in the minor groove. Okay, in the minor groove, the pattern is proton acceptor, hydrogen, proton acceptor. Minor groove for a TA base pair, proton acceptor, hydrogen, proton acceptor. Okay, so again, ambiguous. We can't tell them apart. Also, this is fortunate because, of course, if we look at the major groove versus the minor groove, let me go back. Okay, so here in this figure, you can see the DNA major groove and the DNA minor groove. Proteins, DNA binding proteins, can be accommodated by the major groove much more easily than the minor groove. In fact, an alpha helix, I don't think we've talked about alpha helices in this class yet. If you've taken biochem also, I know Hannah and Emily have taken biochem, we did talk about minor grooves. In fact, we talked about DNA binding proteins too. Alpha helices, which are a right-handed spiral that's made in proteins, they can fit very nicely, almost perfectly, into this major groove. Okay, and then proton donors and acceptors on the alpha helix can line up with proton donors and acceptors on the sides of the base pairs and recognize specific sequences of DNA in that manner. Okay? So take-home message here, the important thing is the major groove contains unambiguous information for recognizing specific DNA sequences, and it's large enough to accommodate DNA binding proteins that are looking for specific sequences of DNA to bind to. Okay, double-stranded DNA can be denatured or melted into single-stranded DNA by heating it to about 100 degrees Celsius. This is reversible by cooling. So this is particularly important if we're talking about, let's say, doing a PCR reaction. So sometimes you need to melt double-stranded DNA. This is reversible. So if we bring the temperature back down to about 37 degrees Celsius, about normal body temperature, then DNA will re-anneal. So it'll, again, two complementary pieces of DNA, they'll line up, form base pairs between the bases, and reform a DNA double helix. Okay, now there's some interesting applications for this. One application I said is we can do PCR reactions by melting DNA, annealing primers, and then extending off those primers using DNA polymerase. Another interesting thing we could do is by denaturing DNA from two different sources and renaturing, we can form hybrid DNA. Hybrid DNA, one application for hybrid DNA is, this is actually a pretty old experiment I did years ago. You can take DNA from two different species, mix it together, and then heat it up. Okay? Then when you bring the temperature back down again, some of those DNA molecules are going to be hybrid DNA molecules between the two different species. And then one strand will come from one species, one strand will come from another species. Okay? And we can look at the degree of hybridization and infer from that just how closely related the two species are. Okay, so if we did something like mix human DNA and chimp DNA, and then denature it, let it renature, the hybrid strands are going to be almost like 99% rehybridized, right? But if we say took human DNA and something like cow DNA and tried to do the same thing, if the hybrid molecules are only going to be maybe, I'm just guessing here, but maybe about 85% double-stranded, and the rest they won't actually reform. Okay, PCR reactions. So again, PCR is an extremely, extremely important chemical reaction that we do in the laboratory. Next week, we're going to be starting our PCR reactions. So I plan on talking about PCR. It's probably a good idea to do it Tuesday before lab. I'll try to make a note to myself. We'll talk about exactly what happens to the PCR engine. You can think of a PCR machine kind of like a Xerox machine for DNA. So we can make lots and lots and lots of copies of one particular part of a DNA molecule. Okay? And in order to do this, it involves melting the DNA or denaturing the DNA. All right, that takes us to the end of the DNA PowerPoint. Do you guys have any questions on this before we start the next one? On anything we covered about the structure of DNA? All right, let's talk about the structure of RNA next. This is a shorter PowerPoint. So I think we should be able to get through this before the end of class today. Oops. Didn't have that set up properly. Just give me a second. Okay, so this is chapter 5. We're going to talk about the structure of RNA. Okay, in many ways, RNA is similar to DNA. Okay, they're both nucleic acids. They're both polymers of nucleotides, but there are some important ways that DNA differs from RNA structurally. So first of all, RNA contains the 5-carbon sugar ribose instead of deoxyribose. Okay? And the difference between ribose and deoxyribose is the presence or absence of a 2' hydroxyl. So let me get my drawing tool again. Cooperate with me, Zoom. Oh, I didn't share the screen. That's the problem. Okay, here we go. Okay, so the first way that RNA differs from DNA structurally, RNA contains ribose instead of deoxyribose. So we know that both RNA and DNA are polymers of nucleotides, and there are 3 parts to a nucleotide. Okay, so just to refresh your memories, the building blocks of nucleic acids are nucleotides, and there's 3 parts to a nucleotide. We have a 5-carbon sugar, either ribose or deoxyribose. Of course, ribose in the case of RNA, deoxyribose in the case of DNA. So we have a 5-carbon sugar. We have a nitrogen-containing base. This is going to be either A, G, C, T, or in the case of RNA, since we're talking about RNA, sometimes U. And a nucleotide has between 1 and 3 phosphate groups. Now, on the 5-carbon sugar, I can't remember if I told you guys in my other class. So we know on a 5-carbon sugar, we number the carbons 1' through 5' in the clockwise direction. So over here, the carbon that's attached to, so we have this glycosidic bond to the nitrogen base. That's the 1' carbon. Here is the 2' carbon. Over here, this corner is a 3' carbon, 4' carbon on this corner, and outside the ring is the 5' carbon here. The difference between ribose and deoxyribose is the 2' hydroxyl. In RNA, we have the sugar ribose, which has the 2' hydroxyl. In DNA, we have 5-carbon sugar deoxyribose, which lacks this 2' hydroxyl. Okay, let's go back to the PowerPoint. Okay, so here is a single-stranded molecule of RNA, and we can see highlighted in red. Come on back. Okay, we can see highlighted in red here is the 2' hydroxyl. So again, counting, here's 1', 2', 3', 4', and outside the ring, the 5' carbon. Okay. So one way DNA differs structurally from RNA is RNA contains ribose instead of deoxyribose. Okay, and again, that's the 5-carbon sugar, and it differs at the 2' carbon. Okay, the second way DNA differs from RNA is that RNA contains the base uracil rather than thymine. Thymine is basically 5' methyl uracil. So if we look at uracil and thymine, they only differ in one methyl group at the 5' carbon, 5' methyl uracil. So where is it? Here. Okay, so here's an AU base pair on top. Here's an AT base pair on bottom. So you can see, if you look closely, you can see the only difference between the uracil and the thymine is this methyl group. Okay, otherwise, they're the same. In DNA, thymine base pairs with adenine. In RNA, adenine base pairs with uracil. Okay, the third way that RNA differs from DNA structurally is that RNA usually exists as a single polynucleotide chain. Okay, so DNA is usually double-stranded. RNA is, in almost all cases, RNA is single-stranded. Okay. RNA doesn't need it to be the template for its own replication. So in DNA, in order to replicate DNA, we know that we need a template strand, and we need to, the template strand serves as, it contains all the information for synthesis of a brand new daughter strand. RNA doesn't need to be the template for its own replication, so it's not restricted in that way. RNA can function as an intermediate, and mRNA is an example of that. So mRNA is the intermediate between the genetic information in the nucleus and protein synthesis, which happens in the cytoplasm. RNA can be an adapter, tRNA. So we talked a lot about how, well, Francis Crick in particular said, you know, this mRNA can't distinguish between different amino acids. So we need some sort of an adapter molecule to act as an intermediate between amino acids and RNA. And it was found that tRNAs do that. So tRNAs can act as an adapter. RNA can act as a structural component, ribosomal RNA. So if we look at ribosomes, they're composed of lots of different proteins, but also some very large RNA molecules. So they can be structural as well. They can be a regulatory component, siRNAs. I'm sure you guys probably heard of mRNA. So mRNA is messenger RNA. tRNA stands for transfer RNA. rRNA stands for ribosomal RNA. You guys might not have heard of siRNA before. Does anybody know what the SI stands for in siRNA? Nobody in class. Anybody in Zoom know that off the tops of their heads? Okay. SI, and let me write this. Well, I'll try to write it down. I'm not very good at writing with a mouse pad. SI stands for small interfering RNA. Oh, I'm having a horrible time trying to draw up this mouse pad. That looks nothing like an E. Okay, you guys get the idea. Interfering, it's INTERFERING. So small interfering RNAs are small RNAs that are complementary for parts of an mRNA, and they can regulate expression of genes by interfering with expression of genes. So siRNA, small interfering RNAs, so that's a regulatory component. Or RNAs can act as a catalyst, ribosomes. You probably learned in other classes that most enzymes are made from proteins. Almost all enzymes are made from proteins. However, some enzymes are made from RNA. And this led to something called the RNA world hypothesis. I don't know if you guys have ever heard of this before. Let me go back to the whiteboard. So when people study the evolution of life on Earth, we have a bit of a paradox. Okay, so let's go back. This is actually something we just talked about not long ago. So around the mid-1950s, Francis Crick proposed what's called the central dogma of molecular biology, which is that DNA can replicate itself. DNA can be transcribed into RNA. And then RNA can be translated into protein. Okay, now this is all well and good, except that in order to replicate DNA, we need DNA polymerase, which is a protein. Okay, we also need, there's a lot of other proteins involved in DNA replication. We need helicases, we need single-stranded binding proteins. So a lot of different proteins go into replicating DNA. Okay? In order to transcribe DNA into RNA, we need RNA polymerase, also a protein. Okay, so we kind of get a chicken and egg, a chicken and egg paradox here if we're trying to figure out how life evolved on Earth, right? So we need DNA to encode the RNA that can be made into proteins, but we need proteins to make more DNA, and we need proteins to transcribe DNA into RNA. All right. Then scientists discovered that some RNA molecules can be enzymes. So some enzymes can be made from RNA. So if some enzymes can be RNA, then RNA enzymes can be used to replicate RNA. Okay, RNA can also be used to store genetic information because of the basic nucleotides. So this led to something called the RNA world hypothesis. So the fact that RNA can be an enzyme solves this chicken and egg paradox by the RNA world hypothesis. So the RNA world hypothesis states that originally genetic information was stored in RNA, and that RNA could act as an enzyme to replicate itself and also to catalyze reactions. And then eventually, DNA, being a more stable molecule, took over the job of storing genetic information, and proteins, being more versatile, took over the job of acting as enzymes and catalyzing reactions. Okay? So again, one of the things that RNA can do is to act as a catalyst or an enzyme. And there's a special word we use for enzymes that are made out of RNA. We call those ribozymes. Okay, so that's like a portmanteau of the word ribosome or ribonucleic acid and enzyme. Mush them together, we get ribozymes. Okay, so we know that almost all RNA inside cells is single-stranded. Single-stranded RNA forms complex secondary structures, including helices, stem loops, internal loops, bulges, junctions, and pseudoknots. Okay? RNA can form some really, really complex secondary structures and even tertiary structures. And actually, this is what allows it to act as a structural component, like if we look at as ribosomes. And this is also what allows it to act as an enzyme if we look at ribozymes as well. Okay, so looking at some examples over here in this diagram, we have single-stranded RNA. So if we have like a single mismatch, like here, that can bulge out. That's an example of a bulge. Here's an internal loop. So this internal loop is made of a short sequence of RNA that doesn't base pair. This is what's called a hairpin structure. So we have a short sequence with base pairing and this small loop that doesn't have any complementary bases. And let me tell you, this is just a small example, a minor example of the types of secondary structures that RNA can form. They can actually be quite complex. Okay, one reason that RNA can form such complex secondary structures is because it doesn't behave by the same rules as DNA. In DNA, we have pretty strict Watson-Crick base pairing. What do I mean by Watson-Crick base pairing? In Watson-Crick base pairing, A's always base pair with T's, G's always base pair with G's. Okay. In RNA, non-Watson-Crick base pairing is possible, and we see it all the time. So RNA exhibits non-Watson-Crick base pairing. We can get GU base pairs, and we can get GA base pairs. These are the two most common non-Watson-Crick base pairs that we see. However, with RNA, all the rules are off. Okay, we can get base pairing with any combination of bases. Oops, I'm sorry, I'm getting ahead. Okay, here we go. Okay, so although RNA is single-stranded, it can form helices. It can form intramolecular helices. So in other words, the single-stranded RNA can fold over on itself and then form localized areas where there are double helices. Okay, again, because this is RNA, it's a little bit different. So the double-stranded helices that are formed in RNA are different from those in DNA. So the 2' hydroxyl on the ribose prevents RNA from forming a B-form DNA helix. The B form of DNA is the normal form of DNA that we see in cells. RNA helices adopt an A form. This prevents sequence-specific recognition in RNA helices. Okay, so let's look at this diagram on the right-hand side. Over here, figure A is bDNA. Okay, bDNA is the form of DNA that's most common in cells. And we can see in bDNA, we have this nice broad major groove over here. We have this shallow minor groove. And again, that broad major groove makes it possible for proteins to get in there and recognize specific sequences of DNA. Next to this, we get aDNA. All right. Now, aDNA, this is found in biology. It's also found in cells, not as common as aDNA. Pardon me, not as common as bDNA. If we look at the major and the minor grooves on aDNA, okay, so here you can see that the major groove and the minor groove are about the same. Okay, it's so aDNA is kind of squashed down, compacted a little bit more. This means that proteins can't recognize the proton donors and acceptors on the sides of the base pairs in aDNA. All right. Now, when RNA forms a local double helix by folding over on itself and then winding into a local double helix, it resembles the aDNA form much more closely than the bDNA form. So this means that it's impossible to recognize, for proteins to recognize RNA sequence by binding to by getting into one of the grooves on RNA. Okay, you can see here it's just not possible. And over here, I should probably talk about this just because it's on the figure. There is a third form of DNA called zDNA. If you look at zDNA, zDNA is a left-handed helix. Okay? And the phosphosugar backbone has to really go through some contortions in order to make this left-handed helix. zDNA is not found in nature. This is just kind of like an artifact that we can produce in the laboratory. So, because it's not really biologically significant, I usually don't talk about zDNA. But just so you know, there are 3 forms of DNA. Okay, bDNA, this is what we normally find in cells. Okay, aDNA, this is found in nature. We find it in cells, but not as abundant as bDNA by far. Okay, then finally and this is also the form, this resembles the double helix that RNA makes. Okay, then over here, zDNA, this is just a really weird thing that we don't find in nature, but we can actually make a left-handed helix from DNA in the laboratory. . Okay, RNA can fold into complex tertiary structures through base triples and base backbone interactions. Okay, so here we see an example of a base triple. Again, this is just another one of the weird things that RNA can do. So we can get hydrogen bonding between three bases to form base triples. Also, we have base backbone interactions. So again, remember that 2' hydroxyl, right? Let me stop the sharing a little bit. So let's go back over here first. So looking at a nucleotide, let's just make this like a U because we're talking about RNA. So we have a 2' hydroxyl, we have 3' hydroxyl, and we have, let me redraw this. Okay, 5' phosphate, and this is going to be connected to the 5' phosphate on another nucleotide. Oops. Okay, so let me draw your attention here. So remember, in RNA, we have a 2' hydroxyl, whereas in DNA, we don't have this 2' hydroxyl. Remember, hydroxyls are polar functional, right? These are polar functional groups, meaning that they can act as proton donors or acceptors in hydrogen bonding, right? So, draw this out a little more. Okay, so there's a polar, this is a polar functional group. We have a polar covalent bond between the oxygen and the hydrogen. So we have a partial positive charge on the hydrogen. So this can act as a proton donor in a hydrogen bond. And we have a partial negative on the oxygen. So this oxygen can act as a proton acceptor in a hydrogen bond. So this is where the base backbone interactions come from. So remember that, so not only doesn't RNA follow the Watson-Crick base pairing rules, the bases can form hydrogen bonds with 2' hydroxyl in the phospho sugar backbone. Okay, so this leads to even more complex secondary and tertiary structures that RNA can make. Okay, let's go back to the PowerPoint. Okay, so RNA can fold into complex tertiary structures through base triples and base backbone interactions. The base backbone interactions were what I was just talking about through interacting with the 2' hydroxyl of the ribose sugar. Proteins sometimes assist RNA in tertiary structures. So I talked about this in Chem 451. So Emily and Hannah, you've heard this before, but there are, of course, the three-dimensional shape or the tertiary structure of proteins is very important, and it's tied very closely to the protein's function. So there are small proteins that assist in folding bigger proteins. And these are called chaperone proteins. So chaperone proteins make sure that that bigger proteins are folded properly so that they can carry out their functions, because their function is tied very closely to the three-dimensional shape. Okay. So there are also, getting back to the PowerPoint, there are also proteins that assist in RNA tertiary structures. So of course, the RNA tertiary structures are very important. tRNAs have an important tertiary structure. Ribosomes, of course, have an important tertiary structure. So there are small proteins that essentially act as chaperone proteins for RNAs in order to fold them into the proper three-dimensional shape. Just like with proteins, the three-dimensional shape of an RNA is vital to its function. So proteins make sure that RNAs are folded properly so that they can carry out their function. RNA tertiary structures are not static. They may have several alternate conformations. Okay, so again, they're similar to proteins in this way. Proteins assume different conformations. Some proteins' activity is regulated by a change in conformation. And in the same way, RNA is not static. It can assume several different conformations. And just like with proteins, this is going to it's going to affect the function of the RNA molecule. Okay, ribozymes. I kind of got ahead of myself. I already talked about the RNA world hypothesis. So let's just read over this quickly. So it was thought for a long time that only proteins could act as enzymes. Considering that proteins are encoded by nucleic acids, what does that say about the evolution of proteins? So basically what that question is talking about is that chicken and egg paradox that I pointed out when we looked at the central dogma of molecular biology, okay, which again is that DNA can replicate itself, DNA can be transcribed into RNA, and then RNA can be translated into proteins. Okay. So in 1967, Ketch and Altman discovered the catalytic properties of RNA. So these were the first enzymes made out of RNA that were discovered. Later, they would be dubbed ribozymes. Again, that's a portmanteau of ribonucleic acid and enzyme kind of smushed together into ribozymes. And this led to the RNA world hypothesis. So again, if RNA can store genetic information, and if RNA can also act as an enzyme, then that explains that chicken and egg hypothesis. If RNA was carrying out these jobs first, and then later on DNA took over the job of storing genetic information, and then later on proteins took over the job of acting as enzymes to catalyze reactions, we no longer have that chicken egg paradox, and it explains how life might have evolved on Earth. Okay. Ribozymes include RNase P, self-splicing introns, and hammerhead ribozymes. Okay, and we are going to talk about introns and hammerhead ribozyme in particular. I've got a couple little exercises we do concerning those two types of ribozymes. Okay. So we got through that PowerPoint. There are a couple little like flash animated exercises having to do with self-splicing introns and the hammerhead ribozyme in particular. I don't want to have to search for them right now, so I didn't think we would get through this PowerPoint this early. So why don't we stop here for today? When we come back on Tuesday, I'll locate those flash animations. You guys, I don't know if you guys who have the book have looked at some of the online content that goes along with this. There's some really cool instructional flash animations. I like to do them together with you in class. Let's plan on doing some of those Tuesday morning before we continue with the next PowerPoint. But for today, let's wrap it up for today. So unless there's any questions from the people at home or the people in class, I'm going to end the meeting here. I know I'm behind on posting the recordings on eCampus. I'll take care of that later this afternoon. Okay, so if there's no more questions, I'm going to end the meeting here. I'll see you guys Tuesday morning.